Hi, everyone. So first I will give a little bit of background why we have a bit of a problem with single cell training. Then I will tell how we try to plug this gap in Finland, what are the European efforts, and what I'm proposing for GOBLET. So at least for us in Finland, the challenge is that single-cell RNA-seq is a relatively new field, so new analysis methods are popping up all the time. So it's difficult to keep up and let alone choose the best, so do any benchmarking. And to make it worse, there are also, in addition to single-cell RNA-seq, there are a lot of other single-cell seqs that are also mushrooming. So you turn your back and there is a new seq already waiting to be analyzed. So all this creates huge need for training. And to make it more challenging, the topic is really a moving target because it's changing all the time. So if we just look at the single-cell RNA-seq data analysis, so at CEST in Finland, so we are the national supercomputing center. So we have we need to provide training at the national level for different universities. So we do run several courses every year. We also have lecture videos available from our past courses as well as other material, of course. And one of the most recent development is that we also set up an e-learning course. In this particular e-learning course, the exercises are done with a web-based GIPS software, so that this course is possible to do also for those people who don't know how to use R or Python, because the focus is on the data analysis methods, not so much on the code. So in this, I will show you shortly, but in this e-learning course, we have lectures both at the beginner and advanced level, and there are obviously exercises and quizzes as well. Anybody can use this course if you want to register so that you can do your final assignments and all that, get a certificate. Then you need to either, if you're an Elixir person, you can use your Elixir AAI, or you can just simply contact us here. So this is what it looks like. So it's just a Moodle-based thing. It's divided in different topics, so that there is the basic pipeline for single cell stuff going from raw data to clusters and marker genes for clusters. And then we also have one section for situations where you have integrated analysis of two samples. And so for each of these sections, you would have several lectures, videos, then instructions on what to do for the practical exercises, and then people go on and fill in some quizzes and so forth. So we use Chipster here, so I just briefly remind you that it's a web-based thing. For single-cell RNA-seq, we have R packages, Seurat and Skater. From training point of view, it's important that it tracks the analysis metadata automatically. So we have actually made ready-made example analysis sessions. So there is the raw data and then the ready-made analysis. So if people want to do self-study, they can compare their stuff. All the course materials are also available, and we provide training accounts. The software itself is open source, and we help with local installations. So this is an example of a ready-made analysis here. So all these are actually files. This is just showing their relationships. Each file would be annotated, so one would know what tool and what parameter used it, and then we can also write notes to them. And in addition, this whole session, we can annotate so that people can do self-study and follow the instructions. So then I move on to the efforts at the European level. So in Elixir, we are setting up a single-cell omics community, or just SCO. So of course, for several years, we have been collaborating in this area, especially on single-cell RNA-seq data analysis training. And for example, already in 2019, we had a big European course. The lecture videos are available. They have had lots of views, over 31,000. But so now we want to sort of consolidate these efforts. And in this community, there are experts, single-cell experts from 17 Elixir countries. We also have connections to non-Elixir initiatives like Lifetime and the Human Cell Atlas. So where we are now, we are drafting a roadmap, which will be then published as a white paper in F1000. And training is one of the big focus areas in this community. Of course, there are other things as well, like data and standards, tools, compute, and so forth, but training is there in a big way. And then finally, what do I propose for GOBLET in near future? So I have had this idea that it would be nice to have a workshop. It can be just a 2-hour Zoom thing for people who are giving training in single-cell RNA-seq data analysis. So we could compare notes, discuss the challenges and best practices. And in order to get you interested, Paolo Czarnecki from NBIS Sweden has made this teaser presentation. You have a link here where he talks about the problem-based learning approach that he has used in the courses. So I leave you here. And yeah, and one more thing. So the idea for this workshop would be like perhaps already beginning of this December. So if you are interested, please let us know. So you can email GOBLET using the email address info@mygoblet.org. So thank you. That's all I wanted to say.